Hey everyone, and welcome to another deep dive. Today we're tackling a topic that seems like it's straight out of science fiction, but is quickly becoming reality: genetic engineering.
That's right, and it's changing faster than ever.
Seriously, remember when computers taking over was just a movie plot? Look at us now.
Yeah.
Genetic engineering is at a similar point. Buckle up because this deep dive is going to be wild.
You got that right. We're talking millennia of history here, from ancient farming practices to cutting-edge labs.
Exactly. We got this really interesting excerpt from Kurzgesagt about the future of genetic engineering, and it's mind-blowing, but to really understand where we're going, we need to start at the beginning. And believe it or not, humans have been doing a form of genetic engineering for centuries.
Way before we knew about genes and DNA.
Exactly. So, before we dive into the high-tech stuff, let's rewind a bit. How about we start with dogs, huh? From wolves to chihuahuas. How did we get here?
Well, it all started with selective breeding. Imagine our ancestors, thousands of years ago, looking at these packs of wild wolves.
A little different from your friendly neighborhood golden retriever.
Right, and they thought, I bet we could make those wolves a bit smaller, maybe less likely to eat the sheep. And so they started choosing the wolves with the most desirable traits to breed.
So, bigger dogs for guarding, smaller dogs for companionship, and eventually, those wolves started looking a lot different.
Exactly. It was basically trial and error, but over many, many generations, they managed to completely transform those wolves. It's like going from a wild stallion to a prize-winning racehorse, all through careful selection.
It's incredible how much we've shaped the world around us, even before we understood the science behind it.
It is. But then, boom, we discovered DNA, the instruction manual of life itself. And that changed everything.
Talk about a game changer. No more blindly mixing traits and hoping for the best.
Exactly. Suddenly, we had this incredible blueprint. We could peek under the hood and see what made living things tick at the most fundamental level. Of course, things didn't change overnight.
Right, we didn't go from breeding dogs to, like, splicing dinosaur DNA right away.
Not quite. But the 1960s were a time of incredible excitement and, honestly, some pretty wild experimentation. Scientists were just starting to understand how to manipulate DNA, and they were eager to see what they could do.
So what were they up to in those labs?
Well, they were limited by the technology at the time. Imagine they were like explorers charting a new world without a reliable compass or map. So they tried everything they could think of. They exposed plants to radiation.
Wait, they blasted plants with irradiation. Sounds a little intense.
It was, but remember, these were pioneers. They were trying to trigger mutations, hoping some of those changes would lead to useful traits.
Like bigger fruits, faster growing crops.
Exactly, and sometimes those risky experiments actually worked. It led to some important breakthroughs in agriculture, like developing new varieties of crops with higher yields or increased disease resistance.
Oh, we've gone from dog breeding to zapping plants with radiation.
Yeah,
Pretty wild journey. But then in the 70s, things started getting a whole lot more precise, right?
absolutely. This is when scientists really started getting a handle on gene editing, specifically gene insertion. Think of it this way. Instead of just shaking up a plant's DNA and hoping for the best, we're talking about carefully inserting a specific gene, almost like a surgeon with a scalpel.
So less genetic roulette wheel
Yeah.
and more targeted genetic changes. This was a huge deal, right?
Huge. It opened up a whole new world of possibilities. We're talking about potentially curing diseases, developing life-saving medicines, engineering crops that are more resistant to pests and diseases. The possibilities seemed endless.
It's mind-blowing. And this is when things get really exciting because in 1974, the world met its first genetically modified animal. Any guesses?
Given what I know about scientists, I'm going to say a lab mouse.
Bingo. A tiny mouse with a huge EG impact. Mice are incredible for research because they reproduce quickly, so scientists can study the effects of genetic changes over generations.
Absolutely. This little mouse revolutionized medical research. We could study diseases in a way we never could before. It led to gene therapies, new drug developments, and a much deeper understanding of genetics as a whole.
From a research perspective, I can only imagine what a game changer that must have been. But did any of this technology make it out of the lab and into, say, our grocery stores? Were there genetically modified fruits and vegetables back then?
Oh, definitely. Remember that super ripe tomato you could find in stores in the 90s? The Flavsauver.
Oh yeah, Flavapsap. It's
Eh.
supposed to, like, stay riper longer.
Right. Exactly. It was genetically modified to stay juicy and flavorful for longer on store shelves. It was a big deal at the time, though it eventually went off the market.
What happened? Were people just not into the taste?
Well, taste might have been a factor, but it also faced some challenges with production costs and distribution. It was a good idea, maybe just a little ahead of its time.
Ahead of its time, yeah. But it really shows how this technology, once confined to labs, was starting to touch our everyday lives. And speaking of pushing boundaries, it's time we talk about three-parent babies.
Three-parent babies. Okay, now we're getting into some serious science fiction territory. It sounds pretty wild, right?
Yeah.
This is where the incredible potential of genetic engineering starts bumping up against some really, really complex ethical questions. Back in the 1990s, scientists were looking for ways to address infertility, and that led to a truly groundbreaking and controversial moment: the birth of babies with DNA from three different people.
Wait, three biological parents? How is that even possible?
It all comes down to something called mitochondrial DNA. So every cell in your body has these little powerhouses called mitochondria, and they actually have their own tiny set of DNA separate from the DNA in your cell's nucleus. And sometimes a mother's mitochondrial DNA can carry genetic disorders that they could pass down to their children.
So if there's a faulty mitochondria,
Exactly. It can lead to some serious health problems. So scientists developed a way to swap out that potentially problematic mitochondrial DNA with healthy DNA from a donor.
so it's like a tiny genetic tweak with potentially huge implications.
Exactly. And it ignited a worldwide debate. Is it ethically right to alter the human germline, even if it prevents disease? It's a question we're still grappling with today, and it highlights the kind of careful consideration we need to have as we move forward with genetic engineering.
It really makes you realize how fast this technology is moving. We started with selective breeding, and now we're talking about changing DNA at the mitochondrial level. It's like science fiction is becoming our reality. And speaking of that, the excerpt you sent us mentioned things like super-muscled pigs and salmon that grow at warp speed.
It's true. Genetic engineering is opening doors we never even thought possible. But, and this is a big but, with that power comes a tremendous responsibility.
Absolutely. Like take those super-muscled pigs, for instance.
Yeah.
Yeah, we can engineer them to grow leaner and faster, but what are the ethical implications for the animals themselves?
Exactly. And what about those fast-growing salmon? It sounds great for seafood lovers, but what happens if those genetically modified fish escape into the wild? Could they outcompete wild salmon, disrupt the entire ecosystem?
It's both exciting and a little terrifying at the same time. We have this incredibly powerful tool with the potential to cure diseases, create more resilient crops, and maybe even help us adapt to climate change. But we need to proceed with caution, asking tough questions every step of the way.
I completely agree. It's not just about what we can do, but what we should do. We need to consider the potential consequences, both the good and the bad. And that's where the conversation gets really interesting and where we all need to be informed and engaged.
Absolutely. So to everyone listening, we leave you with this. What possibilities and potential pitfalls of genetic engineering have you most intrigued or concerned? It's a conversation that needs all of us, because the future of genetic engineering isn't just happening in a lab somewhere. It's unfolding right here, right now. And on that note, that's a wrap on today's deep dive into the world of genetic engineering. Until next time, stay curious.